Hello, I'm Shigeru Hirano at the Kyoto Prefectural University of Medicine Department of Otolaryngology and Head and Neck Surgery. I'd like to talk today about protection of vocal fold by basic fibroblast growth factor. Vocal fold is like a string of the instruments such as guitar or violin to create voice sound, which create from 100 hertz to 800 hertz sound range. And this is a very rapid vibration that we cannot see the vibration with our bare eye. This is the high-speed digital imaging to capture the vocal vibration. The vibration occurs from the inferior surface of the vocal going up to the superior surface. This is a really quick vibration. This is only because the vibrate like this, and this function is supported by the very unique histoanatomy, microstructure of the human vocal. Their structure is the most unique and important part. This is a coronal section of the vocals. The most part is occupied by the TA muscle. This is vocalis muscle or vocal muscle, which provides a good scaffold by contraction of the muscle, and a very thin mucosal surface vibrate from inferior surface to superior surface. And the important point is that this mucosa has a ligament which is called vocal ligament. This is the only mucosa that contains a ligament inside the mucosa, and this ligament is regarded to contribute in maintaining the tension of the vocal mucosa as a string of a high-pitched string. So this is inevitable. This is essential to produce wide range of voice. Hyaluronic acid is also important. This is essential molecule to keep vibration of the vocal folds. If we remove the hyaluronic acid, the vocal fold never vibrate again. Cover body theory represents the function of the vocal mucosa. Cover is the TA muscle, vocalis muscle, which provides the scaffold, and the superficial layer of the mucosa vibrates in rapid format, and the vocal ligament keeps attention. This sophisticated anatomy can be damaged by the vocal abuse, such as in singer or other professional voice users, inflammation, smoking, aging effect, or surgery to the vocal. So vocal can have a hematoma, polyp, a varix, and this is a direct injury by the surgery to the vocal. The worst sequel is the vocal scar, which can caused by inflammation, trauma, surgery, radiation for the cancer even, and this irreversibly disrupts the mucosal anatomy, and severe permanent dysphonia occurs. We have no optimal treatment so far. This case shows very limited vibration, and this case shows no vibration at all. No vibration, no voice. This is a really severe pathology. Histology of the vocal scar of the human vocal fold shows that the whole mucosa is occupied by the thick collagen bundles, which is a fibrotic scar, and there's no layer structure at all, and there's no hyaluronic acid at all. So this is the biggest problem. So we have to avoid this histological change after the injury or damage to the vocal mucosa. Wound healing has only two fates, regeneration or scarring. The scarring is a very important mechanism to protect the wound from bacteria. This is a quick and strong, but if it occurs in the vocal, it causes severe dysphonia. So we lack regeneration of the vocal mucosa. It's not so easy because the wound healing process is very complicated, consisting of three phases: inflammatory phase for 3 days, proliferative phase for 1 month, and remodeling phase up to 1 year. It's a long, long mechanism. There are many elements that affect the wound healing and the wound healing fate. For example, fibroblasts and myofibroblasts are the main cells that create the extracellular matrix after the injury. And it really depends on these cells' function, how it is cured, with scar or less scar. Stem cell is also important, and there are several growth factors or extracellular matrix which affects the wound healing. So it's really complicated and we cannot see everything. This is very difficult to review everything. And now, basically, reactive oxygen species or oxidative stress affect everything, including cells, extracellular matrix, or cytokines and growth factors. So control of ROS should be a great target to improve the workflow and healing mechanism. Reactive oxygen species is naturally produced in the body every day, every site of the body, and it has cytotoxicity which causes cancer, aging, diabetes, ischemia, or others. And during the wound healing, ROS is produced, and it's important to produce ROS because ROS kill the bacteria and protect the wound. But excessive ROS causes poor wound healing with scarring. This is the injured vocal model using rat. The rat vocal is very short, it's like 2 millimeter. Under endoscopy, we cut the vocal mucosa to create an injured model. Histology shows on day 1 and day 3, there is no epithelium and there are many infiltration of the inflammatory cells. And day 5, re-epithelialization is completed, inflammatory cells is gone, and fibroblasts accumulated, and then scarring effect is continued up to 2 months. After 2 months, there is mature scar with very thin and hard tissue. 4-HNE is the marker of ROS, and there are many ROS during day 1 through day 3, but day 5 it's almost gone, and there's nothing after that until 2 months. So these ROS during the early wound healing period is very important for the future of the wound healing. We also confirmed that inflammatory cells produce ROS during day 1 to day 3, so we need to suppress the production of ROS during this period. Basic fibroblast growth factor stimulates the ingrowth of fibroblasts and modified function of fibroblasts in terms of extracellular matrix production. And the good thing is that it increases hyaluronic acid production in the vocal fold. It also has the power of regeneration of vascularity or improved wound healing. And also, basic FGF has proven to have antioxidant effects. This study shows regenerative effects of basic FGF for arteriosclerosis or Barrett's diseases by blocking ROS to improve endothelial progenitor cells functions. So FGF block the ROS, and then the endothelial progenitor cells has more function to regenerate the vessels. Based on these findings, we performed the protective effects of a basic FGF of a rat vocal injury. So this is the same model. The rat vocal was injured, and then basic FGF was immediately injected to the wound site. In sham group, saline was injected into the vocal. We performed PCR at day 7 and histology at day 56. The histology at the 2 months shows that basic FGF 100 nanogram application maintained hyaluronic acid very nearly to the normal side. Statin shows very bad scarring without hyaluronic acid. Contraction of the lamina propria was also better or best in the 100 nanogram basic FGF treated site as compared to the other dose or saline. Collagen accumulation was significantly better in 100 nanogram basic FGF treated vocals. So basic FGF can prevent scarring, maintaining hyaluronic acid, and reducing tissue contraction, and reducing collagen accumulation. Gene expression shows significant upregulation of HAS2 and HAS3 and HGF in the group of 100 nanogram basic FGF treated vocal folds. HAS is a hyaluronic acid synthase, and HGF is hepatocyte growth factor, which has strong anti-fibrotic effects or regenerative effects, which is confirmed in many other organs. So this is a good effect of the basic FGF during the wound healing of the vocal mucosa. In conclusion, basic FGF injection to the injured vocal folds reduce scarring effect by preserving hyaluronic acid, suppressing collagen deposition, and also preserve the lamina propria. Upregulation of gene expression of HAS and HGF were confirmed. Basic FGF is useful for prevention of scarring of the vocal fold, protecting the vocal fold. So, thank you very much for the attention.